Hello, I am Dr. Sukirti Upadhyay. This content is developed by me in collaboration with IITM University. I am Associate Professor there, and my topic is a brief review of various methods of sterilization for pharmaceutical preparation. This content is for pharmaceutical microbiology B Form second year third semester. Next. Sterilization. Sterilization is a process by which deactivation or elimination of all viable microorganisms is achieved by means of different techniques. The need of sterilization arises due to excessive presence of microorganisms in environment, and most of the microorganisms are harmful to living organisms, and in pharmaceutical and industry the contamination with microorganisms can give rise to life-threatening conditions. Therefore, there is a wide classification of pharmaceutical preparation as sterile and non-sterile. Of course, non-sterile is regarded as contaminated, but in case of non-sterile pharmaceutical preparation, they are not contaminated. Only a small or permissible amount of uh, microorganism is present there. Only there is a limit, and this limit is mentioned in various pharmacopoeias, just like Indian pharmacopoeias. Now comes to the topic types of sterilization. There are many types of sterilization. Sterilization can be achieved by the use of heat. Of course, this is not for heat labile material. So, in sterilization by heat is also obtained by dry heat sterilization. This is a universally accepted method for various pharmaceutical but not for thermolabiles. And this process runs, we can perform this process by flaming. In this method, we can use direct heat. This method is used for sterilization of forceps, needles, scalpels, metal spatulas, etc. Second here, there is second hot air oven. This method is for substance which can tolerate temperature up to 250 degrees centigrade. In case of many pharmaceutical preparation, including herbs, etc., which are not thermolabile, we can use hot air ovens. Now comes to micro moist heat sterilization. This can be achieved by autoclaving because here sterilization is performed by steam under pressure. It is an efficient method in destroying wide range of microorganisms. And next one is tyndallization. Another method is this process is also known as fractional sterilization. This was developed by John Tyndall. This requires heating the material at 18 degrees centigrade for 1 hour or at 100 degrees centigrade for lesser time on 3 successive days. Next method is heating with bactericide. This is based on principle that bactericide is more efficient at high temperature. If we use same bactericide at two temperatures, one is high and one is low, its efficiency is increased with temperature. Next is pasteurization. Pasteurization, it is a method, very efficient method to kill 97 to 99% of bacteria, although it is unable to kill spores. It is used in milk industry. Pasteurized milk is always available here. Next is aseptic technique followed by heating. In this method, the material in bulk form is filled under aseptic condition into pre-sterilized container, sealed and sterilized for relative short time. So here, when the container is contaminating the sterilized pharmaceutical, this is not happening here because container is already sterilized. This is an efficient method for pharmaceutical and food preparation. Next method is dry heat above 150 degrees centigrade. This method is for aerosol of bacterial spores. They can destroy at 225 degrees centigrade for exposure of a very short time, zero point 0.4 to 0.6 seconds. Next is Next is cold process. This is sterilization by radiation. This is called cold method of sterilization. Radiation is of both types, electromagnetic and particulate. Destruction of microorganisms occur due to interference of cell metabolism. Next method is ultraviolet radiation. This method is used to inactivate various viruses and bacteria. It is used in vaccine production and sterile filling area for antibiotics, etc. Next method is ionizing radiation. Next is chemical process of sterilization. This require uh, ionizing radiation. This method is used for sterilization of injection and preservation of food. Next is sterilization by filtration. This method is used for sterilization of large volume parenterals and eye drops because the pore size of the filter can checks bacteria, their spores to contaminate them. Next is chemical process of sterilization. This requires activity of various gases. A. Ethylene oxide. It is for sterilization of plastic, paperboard and powder. Ozone. It is protoplasmic oxidant. This is used for sterilization of food and water. Next is formaldehyde. It kills all spores.